On behalf of TCAN, I would like to welcome you to this web event and thank you for joining us today. Our first speaker will be Dr. David Martyr, Chief Executive Officer at TCAN.
Ladies and gentlemen, welcome to our TCAN web event on our next generation Fluent liquid handling family of products. My name is David Martyr, I'm CEO for TCAN, and I'm very grateful that you found time in your busy schedule to join us today. TCAN has been a pioneer in automated liquid handling systems for over 30 years, and we've built our reputation on innovation, performance, quality, customer care and support. Initially with our Genesis platform, then with our Freedom Evo Advanced platforms, we've built a huge installed base worldwide, with systems configured to a very wide range of customer needs and applications. Today we're announcing a further significant step with the launch of our new TCAN Fluent platform and the availability of our first set of applications-focused system configurations. Fluent is the result of extensive voice of customer work and incorporates the latest technology to deliver significantly higher productivity and performance than our earlier generation products. Our new Fluent products are more sophisticated, but at the same time are easier to use and have been configured and the software tailored to excel in specific application areas. On this webcast today, you will especially hear about how Fluent can be used in the very important field of cell biology and cell-based assays. My colleagues are going to lead you through the main features and capabilities of our new systems, and I very much hope that by the end of this webcast you will have a good overview of our new products and how they can be used in your work. But especially I very much hope by the end of this call you've been able to pick up some of the enthusiasm we have at TCAN for this exciting next generation platform. Now let me hand you over to Ian Shuttler, who will introduce the rest of the TCAN team presenting today.
Thank you for joining us today to learn more about the exciting new addition to TCAN's range of laboratory automation solutions, Fluent. My name is Ian Shuttler and I'm the Head of Strategy and Portfolio Management in the Life Science business at TCAN. I'd now like to ask the team who will be presenting today to introduce themselves to you.
Hello, my name is Bronwyn Forster and I'm Senior Product Manager for the Liquid Handling and Robotic Systems.
My name is Susanne Braun and I'm Senior Product Manager for Cell Biology.
I'm Hal Warenberg, Product Manager Software.
We invited you to this web event today to witness the dawn of a new era in automated cell biology research, and that today a new laboratory automation solution would emerge. I'd now like to ask the team to unveil Fluent to the world, a simpler, more productive, more intuitive laboratory automation solution.
Ladies and gentlemen, welcome. Thank you for taking time today to join us for this special event. TCAN is proud and delighted to present the latest addition to its liquid handling portfolio, a new laboratory automation solution. Ladies and gentlemen, please welcome Fluent. In addition to its application focus, the Fluent offers a series of benefits to the laboratory. An outstanding work table capacity combined with a compact footprint in the lab. High speed of movement of robotic arms, parallel movement, increasing productivity, but still maintaining the precision of movement for high density formats or the high precision of liquid handling that our customers have come to expect from TCAN. A new runtime environment on a standard touchscreen interface, which helps the automation of complex sequences be reachable by any scientist in the laboratory. Now I know you're dying to see the system up and running. Just like to show you a couple more things. Firstly, the flexible channel arm, an 8-channel arm where the tips are capable of spacing between different labware formats. This is powered by a completely new liquid system, the CXP pump based on the Centris pump from TCAN Systems. It has twice the resolution, which offers us the ability to go down to smaller volumes with higher precision and maintain a larger volume range over the entire channel. The middle arm here, the multiple channel arm 384. This is unique in being able to change out adapters between 384 and 96 format. This happens automatically and within seconds. The capacity of the work table and the speed of the arm adds together with this head to bring productivity to a new level. And then the robotic gripper arm, a completely new robotic arm capable of taking plates from the peripheral devices onto the work table. It can access also any place below the work deck, making full use of the footprint. It's faster, it's quieter, it's smoother, it's just awesome. So, seeing is believing. I think it's time to see the system in motion, and using the standard touchscreen interface and runtime environment, Hal is going to show you how it all puts together.
Okay, that's my cue. I get the honor of showing you guys the operator interface for Fluent. So on the standard touchscreen, we tried to give all the tools that the operator needs for their daily workflows. We start with something as simple as logging in. I hope our IT department doesn't mind that I am now showing my password worldwide. Once you've logged in, you actually are given the things that you have permissions for. So now that I'm logged in, I'll have the access to the assays that I have the permissions to run, and my favorites will be set for me. And there are two primary functions I can do. There's system care and method starter. So let's just go right to the fun stuff, method starter. We'll start our first assay right here. Some of the other things that we give on the touchscreen for the operator are tools so that instructions can be presented, whether that's simple text or pictures or video. And when these go, we actually have control over the status lights as well. So here we have an example of custom text on the screen and a custom status lamp color for us. As we move through these screens, we can change the colors, we can change the content on the screen, and you can actually put together a loading guide so that the operator knows everything they need to do as they're setting up the run. Here we have a green blinking light. This is actually the same color we have when there's a system pause. Red, this is how we get the user's attention if something unexpected happened during the run. And of course, we would have clear instructions for what to do right on the touchscreen. But I'm teasing you a little bit. We want to see it run. Let's go ahead and do that right now. We're going to start off with the robotic gripper arm fetching a plate from the hotel at the same time as the flexible channel arm is picking up tips and preparing to aspirate from tubes to plates. The robotic gripper arm is fetching a plate from the top of the hotel, and it will deliver it right here for me. Notice it's even going underneath the multi-channel arm. The software is aware of where all the arms are in three-dimensional space and where all the work table objects are as well. This enables us to pull off a couple clever tricks here. You'll notice there the multi-channel arm moved out of the way of the flexible channel arm on its way to the plate. Pay close attention here as the tips come out of these tubes. They stay nice and low on their way to the plate. This is enabled by the software because the software knows how tall the tubes are and what is positioned where on the work table. This allows the software to create a very optimal move path. This is something that we call Pathfinder. So we're taking a very efficient path, and the hardware is moving incredibly fast and incredibly accurate. This makes the entire system have just amazing efficiency. The next thing we're going to show off here is the robotic gripper arm grabbing a plate out of the hotel, and we'll set it down here, again taking the lid off. We're preparing to show you some of the features of the multi-channel arm. The multi-channel arm is a 384 well arm that's actually capable of picking up different adapters that allow it to work with, say, 96 tips, or in the example we're doing here, we just picked up the 96 tip adapter, and we're about to go up and over this stack of tips to the back, and we're going to pick up only 4 rows of tips out of a 96 tip box. And with these 32 tips, we will aspirate from the same plate that we started with. So we pipetted 32 tubes into the first 32 wells of the plate. And with these 32 tips, I will aspirate from those wells and dispense replicates into the other 2/3 of the plate. At the same time, you'll notice that the robotic gripper arm is continuing to take plates out of the hotel and take the lids off and put the lids back in the hotel. This is, of course, setting up for the next little step. Here we drop the tips off into a through-deck waste. This through-deck waste can actually be put anywhere on the work table. That's one of the major advantages of our dynamic deck system. We're completely flexible on where anything on the work table goes, including waste. Here we've picked up 96 tips off the top of the nested tip stack. We're aspirating from the 96 well plate and pipetting into the 384 well plate. This is a really good time to mention that this system is teach-free. And what do we mean by that? Well, the hardware delivers such a high degree of mechanical accuracy and precision that we can actually go to any spot on the work table once the instrument has been installed and set up correctly. We don't have to check any of those positions ahead of time. This move here is one of my favorites. We're actually switching from the 96 tip adapter to the 384 adapter, and we're picking up an entire box of 384 tips all at once. These tips will come to the 384 well plate, and again, after we installed and set up the instrument, all I had to do was put the standard carriers on the work table, put our plate definitions right on, and program the command to aspirate, and then press go. I don't have to do any positional verification or any teaching after that point. So we just pipetted from a 384 well plate to a 384 well plate, and we dropped the tips off, and we get to show off the 384 head adapter also working with a set of partial tips. This time we only picked up 2 columns, so we're actually pipetting with 32 tips, and we can use this to do something like a serial dilution across the plate, which is what this arm is doing right here. The software knows that it's in front of that tip stack next to the active carrier, that there's these high objects around it, and that it can move without any risk of collision. So the software is actually keeping an eye on the work table conditions even as they change. So as the tips get used, it will know that there's more free room to move around the work table. We'll put those tips back and actually show off my favorite arm on the system. This is the robotic gripper arm. Here we have it manipulating a stack of plates. So if you had a compact instrument, say without hotels on the edge or a peripheral device, you could do things like this to save space on the work table and deal with stacks of plates and send them across the work table and restack them up when you need to have more pipetting positions. Or you can take a plate through the work table to an integrated device. Again, with the dynamic deck, you can actually put that gap anywhere on the work table you want. And it allows you to deliver plates to readers, washers, incubators. For some examples of that, you can see that on the website. I didn't have any on hand for the demo today, sorry about that. Now I'd like to show off the advantages of one of TCAN's primary design philosophies. Not only are all of the arms impressive in their individual rights, because we've separated the tasks the arms perform into separate arms and modules, these can all be carried out in parallel. For example, we have the flexible channel arm pipetting from a trough into the tubes. At the same time, the multi-channel arm is doing a serial dilution in a 384 well plate, and the robotic gripper arm is returning plates to where it came from, restacking them, taking lids out of hotels and putting them back on plates that were brought out earlier and bringing those back to their home positions. So not only is each arm incredibly fast and incredibly efficient, all of these actions happen at the same time in parallel and bring a massive benefit to the overall throughput of a system like this in your lab. Of course, these are just tools. What we're really excited about is the work we've done with our partners over the last year to show how these tools can bring incredible benefits in your lab.
You've already seen in the presentations from Bronwyn and Hal the capabilities of Fluent. Especially the capacity and the speed are outstanding. I would like to give you now a brief overview of how your cell-based assay work can benefit from Fluent. We are convinced Fluent is different and hope you think the same in the end of this presentation. We've been working with selected partners to provide complete solutions for you in key areas of cell-based assays. The partners are listed here in alphabetical order, and I would like to take the opportunity to thank them for the fantastic collaboration and the great results we could achieve. The first one in the list is Cisbio bioassays, and they are very well known for their HTRF technology. We've been selecting an Epigenetics assay from their portfolio and have been working with this complex high throughput assay and automated it on the Fluent. DiscoveRx has one of the biggest selections of GPCR assays, and we of course have chosen one of their GPCR assays to show you the versatility of Fluent. European Screening Port is well known for their experience in screening and assay development. In the collaboration with them, we've been focusing to show you a cytotoxicity assay in a very compact footprint and to show you the robustness of the instrument within Thrombin assay. Let me first show you the instrument we've been working with. In this slide, you see the mid-sized Fluent that we've used to develop different applications. You see the different arms. To the left, the 8-channel flexible channel arm, in the middle, the multiple channel arm, and to the right-hand side, between the 1 and the 2, the robotic gripper arm. I want to point out a couple of more elements of this instrument. For example, labeled with number 3, the TCAN carousel that has a huge capacity for different labwares. You can store microtiter plates and disposable tips. Below the carousel, you see an incubator integrated. This is the latest generation of the Cytomat from Thermo. You see on the back of the instrument, labeled with number 1, a 9-position hotel that we've used to store lids. It's a bit hidden behind the stacks of disposable tips. Also a bit hidden by the stacks of the disposable tips is the shaking option labeled with the 2. Just beneath number 2 you see an opening that is the access point for a device below deck, labeled with number 4. A comparable opening is to the left-hand side, just above number 4. The devices that are integrated below deck are shown in this small animation. Number 4 is a hydrospeed that you can use for gentle cell washing. And number 5 is one of our multimode readers, the Infinity M1000 Pro. In this third small animation you see the transfer station of the Cytomat. So you see that the right-hand opening in the dynamic deck is the access point for 2 devices below deck, and that the transfer station of the carousel is sitting just above this opening. That is a really compact instrument that you see here. In the next couple of slides, I want to give you highlights of the applications that we've been developing with the partners, because we have not hours of time. Let us have a look at the first application. This is an epigenetics assay that we've been working on with Cisbio. As I mentioned before, this is a rather complex high throughput assay that we've been doing here. We've been working with compound treatment over several days, and that's why we had for this specific application a HEPA hood installed on top of the Fluent. You see on the right hand side the front page of the application note, and you can always, from all the applications that I'm going to show you now, go to the Fluent web page and download all the application notes. We've been working here with a couple of different cell lines, adherent and suspension cell lines, and they've been plated in different cell densities. Since it is a Cisbio assay, we have of course been working with the detection for HTRF, and we've used one of our Infinity readers for that. The assay is specific here because we automatically swap between the 96 channel head and the 384 channel head, and this is done without any human intervention. The assay is performed in a 2-plate protocol in 384 well format, and that adds to the complexity, and what this means I'm going to show you in the next slide. It is not necessary for you to read everything that is in the graphics. It's really tiny font here, I know that. I just wanted to use that as an illustration so you can easier follow my explanations. Everything you see here in this slide is after the cells have been stimulated, incubated and lysed. So this is just the preparation for the HTRF detection. As you can see on the left hand side, labeled with an A, we're starting from a 96 well plate. If you look at the upper part of this graph now, you see that we transfer with the 96 channel head 10 microliter in 4 aspiration dispense steps into a 384 well plate. Then you only need to add the HTRF conjugate, and then the sample preparation is ready. The upper part of this graph shows you the H3K27 trimethylation part, while the lower part is for the total H3 detection. You see that in the lower part it is almost the same steps that you have to go through, just that we transferring in the first step a smaller volume, which is only 3 microliter. You then add the HTRF conjugate for the total H3, and then you need to fill up the volume in the wells, so you have the same volume in the lower 384 well plate as you have in the upper 384 well plate. The HTRF conjugate transfer is done by the 384 channel head. So you see this is the situation where the robotic instrument just swaps between the 2 different adapters. Looking at the source plate, the 96 well plate, and then ending up with two different 384 well plates for two different HTRF detection modes, you see why this is called a two-plate protocol. Here you see this is a quite complex slide, but I'm going to guide you through. On top in the gray bar, the names of the different cell lines are given. So you need to read from the top down to see the data for the specific cell lines. In the first, so to say, column, you see the data from the SUDHL6 on the left-hand side, the bluish bars, and this is the methylation for the H3K27 trimethylation. Below that, you see for the same cell line, the total H3. What is very obvious here is that we've been working with different cell densities. They are illustrated by the bluish colors, and in the lower part by the pinkish colors. The groups of bars illustrate the timing. The first group is 24 hours, the second group is after 48 hours, and the third group in one graph is after 72 hours. If you look at the upper methylation graph for the second cell line, you see that the bars on the right-hand side are black. This is because we've been working here with a higher cell density that we didn't need to use for the SUDHL6 cell lines. In general, what you see here on this slide is that the methylation level for the H3K27 trimethylation is way higher than for all the other cell lines. And that is, of course, because this cell line carries a mutation in the EZH2 histone methyltransferase. This is actually only the starting point for the experiments to identify the right cell densities to work with in the assay. All further experiments have been carried out with a compound in a serial dilution to show you dose response results. I'm not going to show you these right now, but you find all these data summarized in the application note. Let's look at the second assay. This is a GPCR assay from DiscoveRx, and as I mentioned before, I want to show you the versatility of our instrument using this data. Of course, we've been working with the PathHunter B-Arrestin, and again, to the right-hand side, you see the front cover of the application note. This assay was performed in different plate layouts to be able to compare the results. We've been using Xanthine as an agonist to stimulate the cells, and since it's the PathHunter B-Arrestin, we've been measuring the chemiluminescence. The assay is originally set up in a 96 well plate, but we've adapted it to a 384 well plate. In this slide, I want to show you the plate layout that we've used for the GPCR assay. You can again see in the upper left-hand, on the upper left-hand side, the compound plate. It's a 96 well plate with a serial diluted compound. You see that indicated by the shading in the color. On the right-hand side, you see a 384 well plate and the multiple channel arm that we've used to transfer the compound to the cell plate. Here we use the 96 channel adapter. In the lower part, you see the control plate that we then transferred into the cell plate with a specific feature of the multiple channel head. It is the partial tip function. You can pick up single rows or several rows of tips, and that's what we've done here to transfer two columns of controls, the high and low, into the cell plate. You see easily that we are not using all wells in the 384 well plate here, but with this layout it was possible to get a 12-point dose response curve in duplicates. Here you see the second plate layout. Again, we start with a 96 well plate to the left, and you see easily that the high and low controls are distributed in a different way. We are using now, again, the 96 channel adapter and transferring 4 times from this plate into a 384 well plate, and you see the different pattern in this plate. We're going to end up with a 10-point dose response curve in this plate, but we can achieve now quadruplicates. In the next slide, I'm going to show you some of the data. On the right-hand side, you see 2 times 3 dose response curves. The left ones are from the recordings with 12-point dose response experiments. On the right-hand side, these are from the 10-point dose response experiments. Each graph stands for 1 microtiter plate. And as you can easily see by eye, these are very comparable results, showing you how versatile and robust our instrument is. The Exendine EC50 is very similar to the benchmark values. In the lower left-hand corner, you see a small table giving you the set prime values for each single plate and for each single experiment. And these set prime values are the best values you can achieve for this type of assay. Let's look at the next application that we've developed in collaboration with European Screening Port in this case. We've been using a very compact footprint instrument, and it's actually the one that you've seen in one of the first slides. The cell lines we've been running here are from the National Cancer Institute panel, and they are used to do cancer screening or compound profiling in cancer. The viability measurements of the cells we've done with an industry standard, the cell titer glow assay from Bromica. And therefore, of course, we use luminescence detection, and here we used again the Infinity M1000 reader. This assay is done in a 384-well format, and again you see on the right-hand side the front page of the application note that is available on our Fluent website. The plate layout for this assay looks again a bit different, showing you the flexibility and how easy it is to set up assays on our instrument. The gray areas in the left plate layout show you the areas where the DMSO is prepared. And you see again that we're using the 384 channel head with the partial tip function. We're aspirating from the first column and then prepare in the first half of the 384 well plate a serial dilution. After this, the disposable tips are dropped to avoid any cross-contamination. A new column of disposable tips is picked up, and the serial dilution is done in the second half of the 384 well plate. With this plate layout, you can either have an 11-point dose response curve for 32 compounds, or you decide to have it for 16 compounds only, but then you would have duplicates. Let's have a look at the data. In the lower part of the slide, you see 3 different dose response curves from 3 different cell lines. We've been measuring here the relative growth after 72 hours, and this was plotted against the baseline, which we measured after 24 hours after seeding. The 50% growth inhibition is just shown for one single compound, the gentian violet, and you see the GI50 values in the small table. Again, further data is available in the application note on our website. The last assay was also done in collaboration with European Screening Port and is a fluorogenic biochemical assay. We've been running here 20 assay plates in only 4.5 hours. Again, on the right-hand side, you see the front page of the application note. This is a fluorescence assay, and we've done the detection with our Infinity M1000 reader, and the assay was performed in a 384 well format. In this slide, you see results from the Thrombin assay. You see 4 different heat maps, and you should compare the top left with the top right and the lower left with the lower right. On the right-hand side, the 2 columns are the controls. You can easily see in the animation where I've highlighted a specific area of the lower plates, and that the pattern from the left and the right-hand side is very similar. It's almost identical. And that shows you that we have excellent set prime values for this assay. I'm proud to say that we've achieved an R-square of 0.94 for this specific assay. I've already mentioned in the beginning that the speed the Fluent runs at is very outstanding. You might wonder why this is critical if you look at your assays where you have defined incubation times. So I wanted to give you an idea how you can benefit from the instrument speed. In this slide, you see 8 bars, and each bar stands for 1 microtiter plate. The gray areas are incubation times, and they are fixed. The red areas are the times for robotic actions. So that means it could be transporting a plate, pipetting, or these type of actions. You see very easily that the faster the instrument is, the more can be done in a given time slot that is defined by your incubation. And this is how you will benefit from the speed the Fluent runs at. Of course, signal detection settings are also influencing your timing significantly. But it's very obvious with this chart that you can maximize your batch size with the speed Fluent runs at. I would like to close my short introduction now, hoping I could show you how different Fluent is. We have focused on selected areas in cell-based assay to show you proof data and give you tested configurations for epigenetics, GPCR, and cell health. We've shown you the outstanding quality and how compact the footprint is of an instrument that gives you robustness, versatility, and high throughput. We are very happy and convinced that we can make your complex cell-based assays easy with Fluent. Thank you for your attention.
Thank you for taking the time from your busy schedule and joining us today at this web event. Tecan invited you to witness the dawn of a new era in automating cell biology research, and that a new laboratory automation solution would emerge today. The Fluent Laboratory Automation Solution is an exciting new addition to Tecan's world-class family of laboratory automation solutions. Bron, Hal and Susanna have amply demonstrated that for cell-based and biochemical assays, the Fluent Laboratory Automation Solution provides simplicity, productivity and confidence. With an intuitive touch interface and teach-free hardware, Fluent is simple and easy to use. Faster and with a greater plate capacity than comparable systems, Fluent is more productive. With next-generation liquid level detection and enhanced mechanical stability, you have more confidence in automating complex cell-based assays. All you have to do is just add liquid. Today, with the public unveiling of Tecan's Fluent solution, a new day has dawned in laboratory automation with the demonstration that Fluent addresses many of the challenges in today's busy laboratories. We know that improving productivity levels for complex cell-based research is a challenge. Processes are complex, space is in short supply, skilled technicians are difficult to find, and greater accuracy is always demanded. With the launch of the Fluent from Tecan, that all changes. Now world-class productivity levels are within the grasp of every laboratory, especially for cell-based and biochemical assays. We thank you again for your attention and joining us today. For further information, please visit the Fluent website or contact your local sales representative. Details are available from our corporate website. I would like to thank David Marta, our CEO, for opening this web event, and for Bronwyn Forster, Susanna Brown and Hal Werenberg for presenting today. Thank you for your attention and goodbye.
We do have some time to answer a few questions that have been submitted online during this event. Thank you for submitting them. We appreciate your interest in Fluent. We do not have time to answer all of them, but we will answer the questions via email within the next 2 days. Right now, we will try to answer 4 to 5 of what appear to be the most popular points raised. And here is the first question. Does this new Fluent platform replace the Tecan Freedom Evo now, or will it in the near future? Bronwyn, maybe that you could answer this question.
Well, thank you for the question. The simple answer is no. The Freedom Evo platform will remain a key part of our laboratory automation portfolio offering. It's a well-established industry-recognized platform, and the Fluent laboratory solution adds an exciting new dimension to this portfolio. So both of the platforms have an array of capabilities, starting with the Freedom Evo 75 and now working all the way up to the Fluent 1080. So we can offer any lab the best solution to meet its automation needs.
Thank you, Bronwyn. Next question. Is it possible to integrate detection for high content analysis? Perhaps, Hal, you could respond to this one?
Yeah. As you probably know from our Freedom Evo platform, we actually have a huge library of drivers available to integrate all kinds of different instrumentation. For the Fluent, we will have the same ability to control external devices, but every integration does need to be evaluated on a case-by-case basis.
Thank you. The next question concerns upgradability. I understand that there will be additions to what you have shown today going forward. Will it be possible to have upgrades to an instrument in the future?
Of course. No, that's a little bit of a joke, but actually all the upgrades that we do have planned for the future will be field upgradable and available to anyone who gets a Fluent today.
Next question. A number of people have asked how much does such a system cost? I think Bron wants to answer this question.
Right, this is a difficult question to answer. There really isn't a one-size-fits-all. We've got 3 new Fluent products, the Fluent 480, 780 and 1080. And with different capacities and choice of automation elements, the systems are freely configurable. So really the price is going to depend on the size, the best arm and deck configuration for your assay, if you want benchtop or cabinet, and so on. So really I suggest that you either contact your local sales organization or maybe via this web event let us know how we can contact you so that we can actually discuss needs and requirements and provide you with the specific estimate based on the needs. Thank you.
And the final question. You have focused on cell-based assay applications today. Can Fluent run other assays such as biochemical, high-content assays, etc.? Susanne?
I'm actually glad you asked this question. The applications we've shown you today are of course only examples. We've been aiming to give you from the first day on a very clear idea of the opportunities Fluent gives you. There are many other biochemical assays and applications for which Fluent is an ideal solution. I think you can contact our local sales reps or via the web page get some more information on this. Thank you.
Thank you for your questions. We appreciate your interest and will respond to all of them via email. Once again, on behalf of TCAN, we thank you for attending this web event. Please visit the Fluent website to learn more. We have additional videos, the ability to download application notes, and other information available. Goodbye.